everyone, welcome to Botany Insider, and in this lecture we'll be discussing about the mutations in complete detail. So, as we have discussed that genetic changes could either occur at the large scale or at the smaller scales. So, if the genetic changes are occurring at large scale, they have the formation of the genetic recombination, or the process is known as the genetic recombination. So, we have already discussed about the genetic recombination in complete detail in the previous video. If you have not watched that video, I would recommend you to please watch it for a better understanding of the complete lecture. And if the genetic changes occur at smaller level, they are given the name of the mutations. So, in this lecture, we will be dealing with the mutations in complete detail. So, what is a mutation all about? So, a mutation in simple words could be called as a change in the DNA sequences, and that change is a significant and also there occurs a basic alteration, not a major alteration that we have studied in the DNA recombination in particular. In this type, we have a change which leads to basic alteration in the sequence of the DNA. A mutation occurs when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. That is, as a result of mutation, what happens is there occurs alteration in the genetic message or alteration in the message that have been carried by that particular gene. And why it occurs? Because the gene might have either got damaged or it might have changed in one way or the other. Mutations can result from the DNA copying mistakes made during the cell divisions, exposure to the ionizing radiation, exposure to the chemicals that are given the name of mutagens, or by the infection of viruses. We'll understand about these bifurcations in detail in the future slides. The changing of the structure of the gene resulting in the variant forms that may be transmitted to the subsequent generations caused by the alteration of single base units in the DNA or the deletion, insertion, rearrangement of the larger sections of gene and chromosome. So we can say that mutations are a random change. They are non-directional in nature. We cannot predict what a mutation could actually lead to. They are mostly harmful in nature. They lead to the formation of an alternative form, and that changed form is given the name of the mutant. It is a non-adaptive phenomena. That is, it cannot be adapted by any other gene. It is non-adaptive. If the mutation have to happen, it would occur in any gene, irrespective of where that gene is placed or irrespective of what is the near gene that is present in this particular gene location. Whether it has mutated or not, it does not affect the mutation of the previous gene. So if we look at mutation, it could either occur at the chromosomal level or at the gene level. And in the chromosomal level also, it could either lead to the change in the chromosome number or it could lead to the change in the structure of the chromosome. And if the mutation occurs at the gene level, it could occur because of the change in the nucleotide, and change in the nucleotide could also occur at the same structure and the number level. So let's talk about the types of the chromosomal mutation, that is the mutation which leads to the change in the chromosomal number or the structure. So basically we have 5 types of chromosomal mutations. The first one is the duplication, then we have inversion, deletions, insertions and the translocation. Each one of the type are depicted by their name itself. In the first one we have the duplication. So the type of mutation in which there occurs duplication of any segment of the chromosome. It leads to the duplication type of the chromosomal mutation. The next one is the inversion. So when there occurs inversion, the two sequences or the stretch of sequence between two particular points gets inverted in one way or the other. It leads to the formation of the inversions. The third one is the deletion, in which there occurs deletion of any particular segment of the chromosome. One is insertion, in which there occurs insertion of any particular segment of chromosome to any unknown place. That is, as you can see that this particular segment is inserted to any unknown location. So this is about insertion. Last one is translocation, in which there occurs translocation or the replacement of the segments of or the sequence of the chromosome. As you can see, as a result of translocation, there occurs change in the place or there occurs translocation of the segment of the chromosome. So these are the 5 types of chromosomal mutations. Let's talk about the gene mutations in detail. So talking about the gene mutations, so they are sudden heritable change, that is, they are not predictable and they occur at a very sudden rate. You cannot depict or you cannot predict that at this particular point the mutation is about to happen. They can happen at any particular time. And this term gene mutation was given by Hugo de Vigris. Make sure you remember about this term. So you can see in the figure, we have the normal DNA in the initial place, but because of the mutation there occurs a change in the sequence of the DNA, and this is the result of the gene mutations that we will be talking in this particular lecture. So let's talk about the various types of the gene mutations that could occur. So gene mutations could either be induced or could either be spontaneous. Induced, when there occurs the mutation, when there occurs the gene mutation because of the mutagenic compounds. When it occurs because of the mutagenic compounds, then we could say that these mutations are induced and they result in the formation or the result in the gene mutations. The second one is the spontaneous mutation, that is, they occur on the spot, and these spontaneous mutation could occur either by the help of the replicative errors or because of the spontaneous license. And in replicative error, the replicative error could occur either because of the point mutation or because of the frameshift mutation. What are these? We'll understand about them in detail in subsequent slides. And the spontaneous license could occur either by the help of deamination, depurination, depyrimidation and the oxidative damage. Let's understand about each one of them in complete detail. So the first one is the spontaneous replicative error. So we are studying about the spontaneous replicative error, which could occur either because of the point mutation or because of the frameshift mutation. So it could occur either because of point or the frameshift mutation. In case of point mutation, the point mutation leads to change in one base pair. Point means single. So point mutations occurs or point mutation leads to a change in a single base pair. That is, there occurs a change in a single amino acids, and this could either lead to transition or it could either lead to transversion. So transition occurs when a purine gets converted to a purine because of the point mutation, or when a pyrimidine gets converted into a pyrimidine only. What does this mean? So what are purines and pyrimidines at the first place? So they are a type of the nucleotides base, and these nucleotide bases helps to form the complete DNA molecule by the help of other components also. So transition leads to the formation of one type of purine into the other type of purine, or a pyrimidine into other type of pyrimidine. Purines include A and G, that is, it includes adenine and guanine, and pyrimidine includes 3 type of the nucleotide bases, that is cytosine, uracil and the thymine. Make sure you remember about this. So when a purine gets converted into a guanine, it is a result of a transition. And in case of pyrimidine, so when a cytosine gets converted into thymine or when a uracil gets converted into a cytosine, we would call it a transition. And when there occurs a transversion, so when a purine gets converted into the other form, that is when a purine gets converted into a pyrimidine, that is when A or G gets converted into C, U or T, that is it gets converted into cytosine, thymine or uracil. How they get converted? They gets converted by the help or because of the changes in their molecular structure. There occurs some change in the pattern of their molecular structure which leads to the transition or transversion. So point mutation is a change in one base pair which leads to a single amino acid change, and as a result of single amino acid change there could occur either transition or transversion. Transition is when a purine gets converted into a purine, and transversion is when a purine gets converted into pyrimidine or a pyrimidine gets converted into a purine. So this was about the point mutation. The second one is the frameshift mutation. So in case of frameshift mutation, it leads to many amino acid changes. There occurs no change in the upstream of the amino acid, but all the amino acids at the point of frameshift, there occurs a complete change in the amino acids downstream of that area, and this could occur either because of addition or deletion. So first thing we need to remember is that there is something called as codon. So 3 base pair leads to the formation of 1 codon, that is. And what are base pairs? So we have just discussed there are either purines or the pyrimidines. In case of purines we had A and G, and in case of pyrimidines we have C, U and T, that is cytosine, uracil and thymine. So we have 5 base pairs. So different combinations of these base pairs leads to the formation of these codons. So either A, U, G, C, U, G, and all the different combinations are different codons, and each of these codons have a particular code. They code for the different amino acids. So because of the frameshift mutation, what happens is that there occurs change in the many amino acids. How? So consider we have this particular sequence, that is ATG, CTT, TCA and TCA. Just consider it as an example. We have this particular sequence. So as we have discussed that one codon is made up of 3 base pair. So let's just divide them. So we have one codon, second codon, third codon and a fourth codon. So frameshift mutation, what it does is that consider in the first case there occurs addition of any amino acids. So let's just consider that there occurs addition of a at this particular point. So now what would happen is we have our final changed amino acid sequence as A, T, G, C, A, T, 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 C, A, T, A, C. So we have this particular sequence now. So what happens is there occurs an addition of a particular amino acids. The complete set of the codons have changed. How? Let's just bifurcate them into codons. So this is the first codon, this is the second codon, we have the third codon and the fourth codon, and this one is the next one. So you can see that there occurs change in the codon at the point of insertion. This is the point where a new particular amino acid was inserted, and beyond this point downstream to that all of the amino acids have changed, but there is no change in the upstream. Initially also we have this codon ATG, and after the change also we have the same codon. So this is the meaning of these 2 points, that there is no change in the upstream as a result of the frameshift, but the amino acid gets completely changed, or all the amino acids downstream of this particular point gets completely changed. And same is the case of deletion. So just consider any particular amino acid gets deleted. So consider this amino acid gets deleted. So if we delete this particular amino acid from this area, as a result of that, what happens is all of the amino acids after this particular point would get deleted, but the amino acid upstream to this particular area remains the same. Upstream means to the left of the point from where the initiation have occurred, and downstream is going towards the right of the change point, that is at the point at which the change have occurred. So this is all about the point mutation and the frameshift mutation. So now let's discuss about the spontaneous lysin. Initially we discussed about the spontaneous replicative error, and in that we had the point and the frameshift mutation. So now let's discuss about the spontaneous lysis. So the first one is the depurination and the depyrimidation. In this case, what happens is that there occurs no change in the backbone, only the bases get changed as a result of the depurination or the depyrimidation. And in this case, what happens is, because the glycosidic bonds get broken out of this particular process, there occurs the usage of a glycosylase. Make sure you remember about this particular enzyme. So let's understand this. So consider this is the pattern which we are using. So we have ATAC. Just consider it for an example, and now there occurs a depurination. So now what would happen is, depurination means there occurs a deletion of a purine. And what are purines? A and G were the two purines. And what is depyrimidation? So when there occurs deletion of a pyrimidine, that is either there occurs deletion of T or C. So in this, let's consider we have the mutation and the T got deleted. So what would happen is, we would have all the three sites, but only the T gets deleted because of this particular mutation. So now there occurs an empty site. So this site is given the name of AP site. Why? A stands for absent and P stands for either the purine or the pyrimidine, whichever have got deleted. As a result of depurination and the depyrimidation, we have the formation of the AP site, and also because of the breakage of the glycosidic bond at this area, we have to use there occurs the usage of the glycosylase enzyme. The next one is about the deamination. This is the second point. So in this, there occurs the removal of the amino group, that is there occurs removal of the NH2, and in this, for the removal of amino group, we use the enzyme deaminase. So now make sure you remember about these 4 deamination, because questions have been framed from this area, and we'll be solving one question from the same at the end of this video. So because or as a result of the deamination, what happens is, adenine gets converted into hypoxanthine, guanine gets converted into xanthine, cytosine gets converted into uracil, and 5-CH3 cytosine gets converted into thymine. And in each of these steps, there occurs the usage of the deaminase enzyme. Make sure you remember about them. Questions have been framed, and it is one of the most frequently repeated topics area. Then we have the oxidative damage as the fourth spontaneous lysin. So there occurs the free radical damage nitrogenous bases, that is the free radicals damages the nitrogenous bases in this particular case. As a result of the oxidative damage, the guanine gets converted into 8-oxo 7-hydroxyguanosine, and there occurs the usage of O2 radical. So it is a free radical, and this O2 free radical is given the name of superoxide. Make sure you remember about this particular type also. So we have the mutagens as either the physical mutagens or the chemical mutagens. So if we understand about the physical mutagens, so they could either be the ionizing one or the non-ionizing one. The ionizing one could either lead to the direct ionization or the indirect one. The direct ionization ones are the one that could be either the X-rays or the beta rays or the gamma rays. Talking about the non-ionizing physical mutagens, it mainly includes the UV radiation, and the UV radiations could either cause the dimerization or the hydration. So dimerization mainly occurs of the purine and purine, but if we look at the most common type of the dimerization, it is of the pyrimidine, that is of the thymine. So it is the most common type of the dimerization. Then we have the hydration. So hydration is mainly of the cytosine. So let's talk about the chemical mutagens now. So this is one of the most important slide from this particular lecture, because in June 2020 also one question have been framed from this particular area itself. So make sure you have a good grip of this particular topic. Chemical mutagens, we mainly have three types. The first one are the base analogy, then we have the base modifiers, and the third one are the intercalating agents. Talking about the base analog, so they lead to the copy of a base, that is, as a result of base analogy there occurs a formation of a copy, and it could either be a 2-aminopurine or a 5-bromouracil. So these are the chemical mutagens that lead to the base analogy. So in case of the 2-aminopurine, it leads to AT-GC transition, that is, AT gets converted into GC, and same is the case with 5-bromouracil. It also leads to AT-GC transition. And what is a transition? We have already discussed. So transition was the phenomena in which there occurred either the purine into a purine and a pyrimidine into pyrimidine. So in this, the purine gets converted into a purine and the pyrimidine gets converted into a pyrimidine. Make sure you remember about the difference between the transitions, transversions, purines and pyrimidines, because we often tend to get confused in these small areas itself. The second one is about the base modifiers. So we have three basic type of base modifiers. The first one are the alkylating agents, the second one is the deaminate, and the third one is the hydroxylamine. In case of the alkylating agents, we either have EMS or the MMS. EMS stands for ethyl methyl sulfonate and MMS stands for methyl methyl sulfonate. EMS causes AT-GC transition and MMS causes GC-AT transition. So this is an exception. Why? Because AT-GC transitions are more common and GC-AT transitions are less common in nature. That is, G getting converted into A and C getting converted into T. So this is the exception case. This is the area where you need to remember about the same. Second is the deaminate. HNO2 is the deaminate that could cause AT-GC transition. The third one is the hydroxylamine, and the hydroxylamine causes GC-AT transition mostly. So this is also one of the exception, and in the exam this year question was framed from this particular area. Third one is the intercalating dyes. So all the dyes are the intercalating agents, that is ETBR, acrylidine orange, proflavin, all of them causes the frameshift mutation, and the frameshift mutation could either be because of the additions or the deletions. We have discussed about the frameshift mutations as well. This is one of the most important slide. Make sure you have a good grip of this. Note down the complete table as it is. This is the table that I made in my notes. So just note down the as it is table. It would help you to understand the complete topic and also learn it again and again so that you have a good grip of this topic. So with this, we're done with the complete topic of mutation in complete detail. Let us just quickly solve one question. So the question says that the deamination of the bases is a common chemical event that produces spontaneous mutations. Which one of the following bases will be formed by the deamination of 5-methyl cytosine? So as we have discussed that the deamination of the cytosine, 5-methyl cytosine, leads to the formation of a thymine. So if we remember this table, we could easily find out that B is the correct answer for this particular question. You can see this is a very direct 2-marker question and you can easily fetch out 2 marks from this particular question. So with this, we're done with the complete topic and one previous year question also. So I hope this video was helpful for you, and if you like this video, please do like, share it with your friends, and please subscribe to my channel. And also I'll be coming up very soon with more informative videos as well. So thank you so much for watching and I'll see you soon. Bye.